an accident in further evolution. This is why Crick had this view. Today we know that the known alterations, there are 16 codons where you actually see in blue there are different meanings for those codons in different organisms. Glycine positions in other organisms. So the take-home message is there's a new transfer RNA, which is has the anticodon here is UGA, and it's a glycine tRNA. And as you can see, it is second most important abundant codon in the organism. So this glycine code, this organism now has 5 glycine codons, and not just 4. And I illustrate with this one a consequence of it. And the consequence is that you know all about horizontal gene transfer. But horizontal gene transfer is when you accept the DNA from another organism. But it has to be a lingua franca that the organism and I will actually have the same code. Otherwise, it's not of use to me. So the fact is, if you do such a recoding experiment, it means that the organism is protected in a way from horizontal gene transfer because no one else can use it. So this is one way of isolating organisms as far as translation is concerned. You probably know that transfer RNAs, especially mammalian transfer RNAs, have very many, let's say 30% of the transfer RNAs are modified, bases are modified. And one of the major base positions is in position 37. That's the one position after the anticodon and is before the anticodon. And you can just see that you have a really large variety. I would like to show you just how complex it is to enforce the genetic code. So if you have oxygen here, that uridine, then of course you can see that it recognizes A ending codons and G ending codons exactly the same fashion. But should this be transformed to thiouridine or possibly to selenouridine, then it is different. Then you can actually see that there is far more A recognized than G recognized. And this is actually the basis why you can then get down to A ending codon recognition, distinct from G ending codon recognition. And it's actually the set of codon-anticodon interactions that was missing in the wobble hypothesis. So you can actually change the code by this. And simply to show you how more complex it is, the enzyme, for instance, 2-thiouridine synthase or 4-thiouridine synthase, or this is an enzyme in cysteine metabolism, they actually require iron sulfur clusters. And these are unusual iron sulfur clusters, and they are essential for enzyme activity. So if you would like to really have the code properly expressed, you have to see that even the iron sulfur clusters are properly inserted. I simply want to show you how different, how many different instances come together for maintaining the genetic code. Something else we saw. This is carbon source-dependent genetic code expansion. Acetoholmium aromaticum absent, but it still grows on methanol. So we then said, fine, why don't we go ahead and try a proteomic analysis to see what happens? And this is the final result. There are several ways, of course, that you know by which methanogenesis occurs. So methanogenesis for methanol is really very important, and it's very good, and it's very easy. And this we have in our organism with 21 amino acids, while we are now down here. This is downregulated when we have our 20 amino acids. This is upregulated. So this methanogenic pathway you find with 20 amino acids, this you find with 21. And there is really then a difference in the carbon requirement and metabolism. So why do the synthetic biologists dream about the genetic code? And based on some of the early work by Tom Rajvandari, it's believed that 32 codons are sufficient to encode all the genetic information in genomes. So you can now simply go ahead and resynthesize E. coli or other genomes and decide how many codons you could leave out by replacing multiple synonymous codons with one other one. And this should leave 29 codons for the insertion of additional amino acids. 61 codons are used for amino acids, so minus 32 should be this. But there are great challenges, because to rewire translation by making orthogonal translation systems, you have to re-engineer the transfer RNA, the amino acid tRNA synthetases, and the elongation factors. And this, the cell, of course, would like to prevent under all conditions, because you touch the protein quality control. So reassigning UAG to stop to a non-canonical amino acid is these days done mainly by pyrolysyl tRNA synthetase. So pyrolysyl tRNA synthetase puts pyrolysine on a tRNA, pyrolysine tRNA that recognizes UAG, and then you can make a pile-containing product. And this is what has been done over the last 10 years in many instances, and I'll give you examples. So you have an original orthogonal amino acid tRNA synthetase pair, and this pair, for instance, pyrrolysyl tRNA synthetase tRNA pile, should not interact with the other amino acid tRNA synthetase of the organism, nor the tRNA should be charged by any of the synthetases of the organism. So this is orthogonality. And Pylorez, we now know, is the ideal synthetase for genetic code expansion. So the genetic code can be expanded with Pylorez. This is just a small example of the over 110 non-canonical amino acids that can currently be charged with pyrolysyl tRNA synthetase. And some individual enzymes, just an individual enzyme, will actually charge more than 20 of these non-canonical amino acids and insert it into protein. So I'm referring to something that was just published in a December issue of Nature Chemical Biology. Pyrolysyl tRNA synthetase has two domains, a C-terminal domain and N-terminal domain in archaea. The N-terminal domain is exceedingly insoluble, and therefore we only have structures of the C-terminal domain with and without tRNA. I'll show it in a minute. And we showed already long ago that the C-terminal domain is active only in vivo in the presence of the N-terminal domain. So this then gave rise to the following. So we had a paper in Nature to show the co-crystal structure of a C-terminal domain of the pylorus with the tRNA, and unlike other amino acid synthetase tRNA complexes, this is the enzyme that reaches into the inside of the L shape, and it has a very intricate specialized synthetase tRNA interaction interface, and that I will exemplify in a minute. So we went ahead to look for molecular evolution tools of pyrolysyl tRNA synthetase with PACE and PANSA. PACE is phage-assisted continuous evolution developed by David Liu at Harvard 6 years ago, which is very simple but very difficult to set up and requires complex equipment. And we decided to make a poor man's method out of it, phage-assisted, non-continuous evolution. The methods are based on the phage M13 replication, so this is of course very, very much faster than E. coli. And applications of these methods, and this is the good thing about PACE and PANSA, which is different from MAGE, which is being used by many of the synthetic biologists and genome engineers to do genomes. This does not require any prior structural knowledge. Otherwise, you need to know you would like to mutagize this because this happens to be the active site or half the active site or whatever. So you can do this. And in this particular case, we showed that both methods give very similar results, and we have isolation of pylorus variants up to 45-fold increased catalytic activity, and mutations are located in an N-terminal domain. And just, I won't show you a slide. When you do nowadays super phylogenetic analysis of the many, many sequences there are, you can actually find organisms where, in contrast to what I told you earlier, there you find C-terminal domains that are active alone without N-terminal domain. And we now have gone ahead to in vitro, by molecular evolution, recreate such enzymes. So this then is a model of the full-length RNA because we were absolutely insistent, and Tateki Suzuki tried everything there is for several years to come up with a structure, because I tell you, the PANSA and the PACE mutations all were in the N-terminal domain. And since nothing was known about the N-terminal domain, we finally had to get to it. So we had a crystal structure that required zinc in it, which we didn't know before. And then we could do it. So the N-terminus binds here, the C-terminus binds here, and these somewhere, there are another 150 amino acids in there between the N-terminus and C-terminus. So it looks as if the synthetase wraps around the tRNA. It's totally different from any other amino acid synthetase tRNA complex. It has the largest interface area for known synthetase tRNA complexes, and you can see, of course, pylorus does not recognize the anticodon, which is a godsend for all synthetic biologists, because they can then use this for any codon that they would like to do. And this is just because it looks very nice. The wild type structure, and this is a mutant structure, and you see how the tRNA binds. In the mutant, where we get the increased activity and different specificity, opening up the structure decreases the tRNA pile binding and leads to increased non-canonical amino acid recognition, but with lower catalytic activity. How many more minutes? Fine. So a different point is recoding UGA from stop to selenocysteine. Most people do not recognize that selenocysteine, even though it's a genetically encoded amino acid, has no amino acid tRNA synthetase to charge it to tRNA. This is done in an RNA-dependent fashion. Furthermore, This reaction, incorporation, is not competed for by elongation, also by release factor 1, but by release factor 2. Therefore, a very different model of protein synthesis is happening. And there is cell B, this new elongation factor is required because tRNA-seq is the longest tRNA, has up to 9 base pairs in the acceptor stem. And there's furthermore an RNA element, at least in bacteria, that is immediately adjacent to UGA. And because the RNA element is within the coding region of the protein, of cellular protein, you can't really do any mutagenesis like you can do with other amino acids, because if you move the anticodon, you would also have to move the sequence here. And if you do this, then of course you have obliterated immediately 20 amino acids of your enzyme. So we developed tools that I don't want to describe here to do this and to make any cellular protein that you like in E. coli. But we then did one other experiment. And this was really the absolutely enthusiastic involvement of Marcus, Joan, and Patrick O'Donoghue, where we wanted to work with formate dehydrogenase. And formate dehydrogenase is the only cellular protein in E. coli, and position 140 is cysteine. And so formate dehydrogenase messenger RNA has here the UGA codon, and the tRNA-seq has the bound to cell B, the UGA, the anticodon that recognizes UGA. So what we really wanted to do to see how many nucleotides in E. coli can we recode by making 64 different codons here. So this is a typical synthetic biology experiment where after creating the right strain, we had to synthesize 64 messenger RNAs with 64 different codons in this position. Then we needed 64 tRNA cysteine variants with 64 different anticodon sequences in this position. And then the secret, of course, you have to find out, and we have always been very fortunate in this, that we get a very easy genetic test that is exceedingly sensitive. And a readout for formate dehydrogenase is benzylvalogen. It's an old test that gives a purple color. And if you look, there are 60 out of 64 codons which you can recode. But then you really have to do it by a chemical experiment, learning from Govind. So you have to purify each FDH from each experiment, determine the enzymatic activity because only SEC-containing enzyme is active, and then determine the SEC content by ICP-MS and identify the position with LC-MS-MS. So with all of this, then we showed that 60 out of 64 codons can be recoded cellular cysteine, some of them completely, and given the fact that they are now, if you then have these sense codons to do this, they are no longer hindered by any release factor. You can get protein increases up to 12 or 15-fold even. And we also found out that some sense codons may even be recognized by RF2, and this is a different analysis that needs to be done. Then I would like to give you a different kind of recoding. And this is absolutely opposite to everything that you have seen in literature by the genome engineers. And this goes via the supplementation-based incorporation methods. So if you have an isostructural non-canonical amino acid that tricks the amino acid tRNA synthetase to be charged, you then get protein variants where you get the analog in instead of the wild type. So it's a natural substrate tolerance of the endogenous host synthetase allows this. And in this particular case, the aim was to replace tryptophan with cyanopyroalanine in protein synthesis. So the 6-membered ring that you see here of tryptophan would actually be replaced by the 5-membered ring with sulfur in it. And to make the long story short, this was simply done by a serial transfer experiment in batch culture, which after 280 transfers and doing some miracles with what you give the strain to grow on, you got a proteome-wide exchange of tryptophan for this thionylpyroalanine E. coli. The cell itself grows only 10% slower than wild-type E. coli. And so basically this has happened. And in essence, and I know the synthetic George Church and others are upset at the nomenclature, we have recoded 20,899 UGG tryptophan codons in this experiment by simply restricting the amount of tryptophanol tRNA tryp, or what that the cell has that can be used. And we are continuing these experiments with analogs that are far more distant from tryptophan to see what the plasticity of the proteome might look like. And I will give you the last experiment, and this was the question, how is a genetic code expressed in nature? This really has always been an interest, because when we in Corrales' laboratory deciphered what the code is, many observations were made and many ideas were embedded in this that really were dogma and that couldn't be challenged. And the question was, is this really so? So can a vast amount of genomic and metagenomic sequence data provide information? We could, and we looked at over 10 terabytes of sequence. And the tRNA sequence structure is the key. And this was work done by Takahito Mukai, together with people from the Joint Genome Institute and Walnut Creek, California. And we took tRNA-seq as first attempt with a covariance model because it's a long and an unusual tRNA. And then one was just trying to sequence. Sorry. So the major point is in here. This determines the anti-codon diversity of tRNA-seq. We got a vast number of sequences. There are many exciting examples that have come out that are different from what I'm going to show you here. But this is what we got for real selenocysteine. So we saw that these are codons that are used by various organisms for selenocysteine. And the take-home message is that selenocysteine in microorganisms, even eukaryotic microorganisms, is encoded by 10 sense codons and 3 nonsense codons. So the idea that you have a firm assignment is out the window. And one reason is, why is there selenium? And you can ask me later. One can speculate for a long time. If you have a protein that has a selenol moiety in it, it's oxidized very easily. If you have a protein that has a SH moiety, cysteine in it, it's also oxidized. The big difference is, with any sulfur reagent, you can reduce the oxidized selenoprotein, but you cannot reduce the oxidized cysteine-containing protein. So if an organism experiences an oxidative habitat, it may actually change and encode for cis-selenocysteine because it will help the organism to survive. This is just one example. So with this then, I would like to leave you with that the likelihood of discovering a new amino acid present in all 3 domains of life is exceedingly low, I don't think it's going to happen because we analyze, sequence so many already. But a new amino acid localized to a small group of organisms is possible. And this is of interest, like with pyrrolysine. So if you need a certain pyrrolysine evolved because a certain enzyme that takes up trimethylamine that is good for the energy metabolism of the cell, that enzyme is pyrrolysine in the active site. The exciting thing is when you think about it, once this happened, the organism could use a pyrrolysine for anything. So you find now organisms that are no longer requiring pyrrolysine for a particular enzyme, but they just use it, and this is really how an amino acid comes into existence, so to speak. If a new translational scheme evolved, nature will use it anywhere when needed. 15 or 20 years ago, we made big discoveries that we said are only in archaea, and everybody said it's absolutely correct. Archaeal protein synthesis is very different from eukaryotic protein synthesis. But then turn around and look at it now, it's not. So there has to be a need for deeper understanding of cellular metabolism, because the relationship of code and use and organismal fitness is something that will determine how many of these engineered organisms can be made. Code evolution is possible. The fitness of organisms containing additional amino acids will be decreased. But the take-home message, the genetic code is far more flexible than we previously thought. And if anyone of young people here give my lecture in 15 years' time, it will be different again. So I have to thank all my colleagues. These are the people in my lab: Anna Černković from Croatia, Felix from China, Kyle from Canada, Huisi Kok from Singapore, Sergei from Russia, Takahito from Japan, Noah Reynolds from the US, Anastasia from Russia, Tateki Suzuki from Japan, and Oscar Vargas from Venezuela. I was fortunate to collaborate with Nedilko Bodiza from Berlin, David Bryson and George Church from Harvard, David Liu from Harvard, and Nick Kilpidis from the Joint Genome Institute, and my former postdoc, Eric Wang, who is at the Academia Sinica in Taipei. And I acknowledge happily support from Department of Energy and National Institute of General Medical Sciences. Thank you.
Okay, thank you, Dieter, for a really enlightening seminar. I think we have time for a couple questions.
Dieter, with highly evolved organism that uses the tryptophan analog, have you done the genome sequence or something like that to see what's the big change, or what is responsible for this now?
So the question is really, do we know anything why this organism is still happy? To our amazement, there are not more than 150 mutations in it. There are 2 mutations in TRPRS to make the TRPRS better. But we also have got some insight that hopefully we'll have in the future, that there are certain gene changes which will make this evolution 10 to 15 times faster.
So if you add the
In this case, it is not toxic. For old people have memories. In 1983, there was a paper in PNAS to show that certain bacteria, B. subtilis bacterium, had fluorotryptophan in the DNA. And this leads to Tom's question. In that case, it was shown that tryptophan was detrimental for the growth of these organisms, because only fluorotryptophan was accepted. And I will tell you then one other story, and I forget for the audience, I forget the name of the person. If then a synthetic biologist comes along and does an experiment, he or she should be cleverer than nature. And this is what we learned from Gobind, what the synthetic biologists did not learn from their advisor. Because Gobind said, whenever you get a chemical, you analyze and look what else is in it. And so there was a paper published in 2005 to regenerate this fluorotryptophan evolution. And the people did glorious work and even put in a paper that the fluorotryptophan had 80 micromolar tryptophan in it. And all the results, of course, were just described, gave wild-type E. coli. Wild-type E. coli, because E. coli knows how to distinguish tryptophan from fluorotryptophan. So this is something that I think in modern times we don't teach students anymore. Gobind insisted that you are responsible for the compound that you use. And I don't want to go into examples. This is absolutely essential. Today you buy a kit, you assume that the kit is fine, and that's it. Sorry. Marshall?
Thank you very much. I have small two queries. With 3.8 billion year history, with all permutations and combinations available under different sets of conditions, which have changed tremendously in every eon, nothing out of this possible combination has been left as a fossil in any of the organisms. They have not been able to carry that one. And do you expect that under the remotest environmental harsh condition or something, you are likely perceive that you will encounter such a thing somewhere? And second part is that two new codons, do they connect with this part under the synthetic biology story?
These are very, very good questions. I mean, what I, as a simple chemist, didn't realize, that in evolution, you come to a fork, and this organism just grows infinitesimally better than that organism, but then evolution goes this way. So you only have a very, very small difference to change. Yes.
It does not have a direction.
So
it goes towards simplicity.
This is correct.
This is after 100 years, Homo sapiens won't be able to synthesize glycine.
Yes.
Yeah. And the codon of this one will become redundant. Okay.
So you mentioned the environment is very important.
Yeah. Agree.
And the environment will drive it.
Yeah. But nothing combination of this one is available in any relic form, any fossilized form, or any of the, in this solar system, which is mine.
I think you or I, we are very likely old enough to look for things like this. They're considered crazy ideas by people. And I will give you one personal. I go to the gym with Tom Stites, a Nobel Prize winner, and I asked Tom, I said, Tom, I think glutamine is the last amino acid. Can you imagine what life would like without glutamine?
Without?
Glutamine.
Okay.
And he said, this is an idiotic question. Don't you know how important glutamine is in protein structure? So I said, yes, today, but heaven knows.
Not much.
Yeah. So I think your questions should be asked.
Okay, any other questions? Yeah.
I have a few queries. The first one is your demonstration of incorporation of tryptophan analog at a specific position. is not that amazing because we know in proteins, tryptophan from the structural point of view, it has very little functional role as such. So mostly it's in the core.
This is true. This is why we use tryptophan.
Yeah.
On the other hand, and this is what my NIH program director calls it, but Dieter, look, there are so many papers show that these 3 E. coli proteins have essential tryptophans in it. So we are actually doing the structure of these proteins, but it will be very likely, as you said.
Because it's most likely the distribution is in the core and the packing,
Yeah.
right? Because in protein crystallographers often do use auxotopic strains and incorporate selenomethionine for phasing experiments. So we can take a auxotropic strain of E. coli, which can't manufacture methionine, and put selenomethionine. All the methionine distribution is quite much on the surface, and so which can be problematic. Sometimes it's a problem. Another question is related to your N-terminal domain. I'm very curious. You wrote in the slide insoluble. Is it insoluble, or you want to infer unstructured?
The main pyrolysis, let me go back one. Pyrolysyl thionosynthetase is an archaeal enzyme. So the organism it comes from has 1.1 molar sodium chloride as a normal concentration in it. In high salt, we can purify it easily. If I go to below 0.8 molar, it just precipitates. So when I said insoluble, it is in our laboratory conditions that everybody uses.
So you give that, you can express the domain independently, N-terminal domain, and put in those conditions. And you can look at least the solution structure of it.
We did it and we got the structure. We got the structure and it helped that we actually put zinc in it. Because we had no idea that it was a zinc protein. Thank you very much for your comments.
Okay, I think it's time to move on, especially since we've now established that as you grow older, you get crazier, but you can still do great science.
Can I ask one question from sir?
Okay.
Ma'am, thank you, sir. So my query is that you told that the SH group in that is because of cysteine in proteins. So is that SH groups can affect the expression of the protein or their solubility? If a protein having a high SH group, higher number of SH group, that is a cysteine, does it affect its expression level, or I should say, secondly, its solubility?
We do not know. And today, of course, you also have the availability of other expression systems than E. coli. So I'm sure if you have a very difficult problem you would like to express it, you can find systems where you can express it. For us, it was simply established methods, E. coli, and we have not looked.
Okay, thank you.
Okay, let's thank Dieter once again. All right, so our next speaker is Umesh Varshney, and he's from the Indian Institute of Science in Bengaluru, and his title is Pioneering Round of Initiation Complex Formation, Triggers Ultimate Steps of rRNA Maturation in E. coli.
Good morning, everyone. It's really wonderful to be here. Meeting a lot of people here from the old time. I'd like to thank Tom and Rajinder to put it together, and the Team India to really do a marvelous job that we are all enjoying here. I joined Khurana Tom group towards the end of '88, December '88, and I was there until July '91. And here is an undated picture that I fished out from my computer last night. I think it must be 1990, because I see here Nripen Mandal. I think he joined a little after I did. And so I think the picture must be 1990, and you can see Sriram here. I can't see from here. I think somewhere here it must be there with his beard on. We listened to him yesterday. Of course, Govind and Tom. Julia is over there. Anne-Marie is also there. So Anne-Marie was really the person taking care of everything in the lab. There is another picture I have. This is in the new building, and that must be in 94 actually, because I was visiting 95, no, we moved in 94, I think. That's the time I was visiting, and I happened to be there around the time that, group pictures are taken, so I'm right there. So again, I think, Ramesh was there at that time, so I guess, it must be around that time. There are a lot of things, I've learned from both Tom and Govind, and really, they've really shaped my life. Before I came to MIT, I knew how to do science, didn't know why I was doing it. But, coming to the group was really wonderful because, one could start asking questions, that would really mean something, people would find them interesting. And so I think, thank you very much, Tom, we don't thank you enough. So it's really, it was lucky for me to be there. And, small encounters with Govind were also very useful. He made me realize what science is. One day I was in the kitchen making my coffee. Govind walks in. He asked me, what I was doing. I said, I'm making mutants. Well, that's our culture. So really, I think the way he was looking at things was really quite, all into science all the time, and that's really changed things. So I think in that year, I also sort of, this is a picture in a restaurant, actually, somewhere close to Tom's home, and I think, so it was really nice to visit at that time. And then a few years later, we had a symposium for Tom that was in 2011, and many of us were there at that time. And so thank you once again, Tom, to let us have that. In fact, I think Asim yesterday made a proposal. I'd like to second that in public. So with that, let me now begin the talk. And what I'm going to do really is some of the slides that you're going to see are going to be the slides that I've used in my Monday seminars. And also in Thursday seminars, because Govind used to have Friday lab group meeting, and Tom was having them on Thursdays. So a lot of these slides are going to be common. And this particular one is really one of the very favorite ones, and I'm not sure what Tom is going to have in his talk also. But this slide really tells that in the cell, there are two kinds of tRNAs. One is really the garden variety, I would call it, elongated tRNA, which is amino acylated. Then with the help of elongation factor TU, it goes to the ribosomal A site. Whereas the other one, which is a special tRNA, the initiator tRNA, is amino acylated with methionine tRNA synthetase. And the formulated tRNA then goes to the ribosomal P site. Now, this is the only tRNA, in fact, that binds directly to the ribosomal P site. All other tRNAs must first bind to the A site here and then move down to the P site. So what that really means is that there are a few questions that can be right away asked from this observation. And that is, how is initiator tRNA selected onto the ribosome? And secondly, how does it participate in initiation process? Now, this is the initiation pathway that has been known for some time. And what is really happening is that much of the important activity in initiation is happening on 30S ribosome. So you have all the initiation factors bound there. Initiator tRNA is there. 50S subunit joins. And then something happens. All of the initiation factors go away. Initiator tRNA occupies the right place. And we have elongation competent 70S ribosome. Now, much of the things that are here in this black box are not really that well understood. And the point here is that I'm trying to make is that through all of these stages, initiator tRNA is right there. So one could, in fact, even use initiator tRNA to understand each one of these steps. So this was the idea that I had when I came back. And of course, we're using the initiator tRNA. So let me tell you something about the initiator tRNA. In E. coli, there are 4 initiator tRNA genes. 3 of them are at 63.5 minutes, and they're part of a single operon. The 4th one is a little away, 71.5 minutes, and there's a single tRNA gene. All of these tRNA genes, in case of E. coli B, in fact, are identical, but K, there is a single change here. But without worrying about that change, the overall structure of the tRNA is shown here. And there are 2 parts that Tom had really established to be very, very crucial for this tRNA. One in the acceptor stem region, which is important for the formulation of the tRNA. But of course, formulation is not there in archaea and eukaryotes. The other region is the 3 GC base pairs, consecutive GC base pairs, which are really highly, highly conserved in all domains of life. And so this turned out to be something which one would want to follow a little bit more to understand how proteins or how initiation happens. Now, one of the things that I did in Tom's lab right in my beginning when I joined was to set up this in vivo initiation as a system. And the SS system, in retrospect, is very straightforward. We have the reporter gene, chloramphenicol transferase. All I had to do was change the start codon from AUG to UAG and make a corresponding change in the anticodon of the tRNA, initiator tRNA. And this tRNA would now initiate from UAG and would confer chloramphenicol resistance. Now, no surprises that when we touch the 3 GC base pair and change the sequence to the sequence that is found in the elongated species of methionine tRNA, the tRNA, of course, does not work here. So here is the system where we have our reporter CAT gene, and this is the tRNA gene where the 3 GC sequence has been mutated, even though it is still called here 3 GC. Genetics nomenclature is somewhat strange. So when we say 3 GC, it really means here that this is mutant, and it does not grow on chloramphenicol. So basically, the tRNA is not functional in initiation, and we of course know that the tRNA is amino acylated, and tRNA is also formulated. It binds to IF2. Everything is fine. It just does not initiate. So this is where I started my sort of work in India, and the work was again going back to very simple methods. In fact, this is another lesson that Tom gave me, and that was that, Umesh, you go to India, you do anything. If you see a growth on the plate or whatever, next morning, that should be a very good experiment. And this is really what I started out with, something very simple. And this was really facilitated by postdoctoral fellow Dinesh in the lab. And what I thought I will do is I'll talk to you about a little bit of these 2 strains. But these are really the suppressor strains. What this assay system that was set up was that we know that when we have mutation in the 3 GC base pair, it does not initiate from UAG, and the cells are actually chloramphenicol sensitive. But now if we make mutation in the E. coli genome, and if this mutation is able to rescue the defect of 3 GC base pair, it would start from UAG start codon and confer chloramphenicol resistance. All we had to do was rule out some of the unwanted mutations in the genome, and then select for the ones which require this mutation, this tRNA, and this reporter. When the 3 things are required together, we have these strains. Now, we've been working on these strains for last many years, really. And we have learned quite a lot about the process of protein synthesis. And what I can give you a little bit summary here is that if we look at why formulation is needed in case of initiator tRNA, there are two things really. One, it of course facilitates binding of the initiator tRNA to IF2. It increases the affinity of initiator tRNA greatly for IF2. The second thing, we've been able to sort of connect it is that there is actually a connection between the formulation and the 1-carbon metabolism. This has been told to us by our suppressor strains. So 1-carbon metabolism, if students would remember, it gives rise to 10-formyl tetrahydrofolate, which serves as a cofactor for formulation. And the 1-carbon metabolism also makes methionine. And therefore, both formyl group and methionine. So formyl methionine is really important. And you would want to initiate protein synthesis only if your cell is in a good energy state. So I think it's a really good checkpoint to start protein synthesis only when the cell is rich. The other thing that we've been learning about is the role of 3GC base pairs. One of the things that 3GC base pair does is as follows. When the initiator tRNA actually binds to 30S, it is not really binding in PP state. It is binding in a PI state. And for it to go from PI state to PP state, it can actually fall off the ribosome. What we realized is that the 3GC base pairs are important for this transition from this PI state to PP state. And we also realized, based on some of the additional work from the suppressors, of course, that the 3GC base pairs are also important in ejecting the initiation factor 3 from the ribosome. And so once you have those 3GC base pairs, you can make a 70S initiation complex where the tRNA is in correct orientation. You can then also eject the initiation factors from the 70S initiation complex to give rise to 70X elongation competent ribosome. So 3GC are really very important. So much of the importance of formulation seems to be right in the beginning of protein synthesis or initiation, and the importance of 3GC base pair seems to be towards the end of or in the later stages of initiation. So I mentioned to you about those D4 and D27. A graduate student, Suman Kapoor, was working on those. And this D4 and D27 turned out to be a mutation in the promoter region of the initiator tRNA genes, where in the locus which codes for 3 tRNA genes. And with these mutations in these suppressors, you can see the promoter activity goes down to really nothing. And if we look at the levels of initiator tRNA in these strains, we find, now this isn't a mistake actually, she overloaded these two lanes to show you that there is a little bit of FMET1 is still made. So this locus actually makes FMET1, and so this is the wild type strain. You have roughly 3 times the tRNA compared to this one, but in these strains, FMET1 is nearly gone. And we can sort of simulate these conditions by making a knockout of FMET1, where you see that the top band is missing, or we can also make a knockout of the second locus, where the bottom band is missing. So she actually did all of this, and she also showed that if we have this strain where the 3 tRNA genes have been deleted, this strain grows all right at 37, but when we subject it to 22 degrees, it doesn't grow that well. The wild type strain grows, but the mutant does not grow. Now she finished her PhD at this stage, but luckily another graduate student, Sunil, a very bright student, joined, and he found this observation quite interesting. He found it very interesting because, often the cold sensitivity is associated with a defect in ribosome biogenesis. And so ribosome biogenesis really occurs, it starts very early on. So we have a precursor transcript which has all of these, regions in the transcript. And as the transcription goes on, these transcripts are cleaved off. You have extensions to 5' and 3' of the 16S, which gives rise to your 17S rRNA. and that is then finally processed into the 16S rRNA. There are many ribonucleases that are involved, which are sort of listed, and I'll keep coming back to these. So you have then finally the mature 30S ribosome. So if we were going to probe for whether there is a defect in ribosome biogenesis, what he wanted to do was look whether there is accumulation of 17S rRNA. Now 17 and 16S migrate very close together on agarose gel. So what he did was he designed oligonucleotide probes which would anneal in the immature region of 16S, that is to 17S or to 16S or to downstream of the 16S. And he can then do the northern blot analysis, and one thing that you can very clearly see is that when you go down to 22 degrees, you accumulate a lot more of the precursor. And he can then take the ratios and come up with this kind of a plot where the wild type strain has that much precursor. As you delete the tRNA genes, the precursor levels go up. At 22 degrees, this really goes up. So he set it up as an assay system for him, and then he started asking questions as to what happens. Now, another observation that he made was that if in the same kind of, if he takes the wild type strain of E. coli, and if he actually overproduces the tRNA, which has mutation in the 3GC base pair, that also gives him the cold-sensitive phenotype. And he can rescue this cold-sensitive phenotype by overproducing a wild-type copy of initiator tRNA. And so this also sort of went back at that there is again a biogenesis defect here. So whether we take away the wild-type copies of initiator tRNA, or we overproduce a tRNA which is defective in the 3GC base pair, he gets the same phenotype of cold sensitivity, he gets the same phenotype of ribosome biogenesis defect. Now, in genetics, you would also then want to combine the two and look at whether there is a synthetic effect, and this is precisely what he found. So when he takes the strain which is deleted for 3 tRNA genes, and he overproduces the mutant tRNA, he doesn't find any growth at this one. So they were working together, and in fact, he can measure that also. So that clearly said that the two are working, doing the same sort of things. Now, one would have wanted to work with the situation when we delete the 3 tRNA genes and overproduce 3GC base pair, but that's a very, very tough situation. The bacteria doesn't like it, keeps coming back with suppressors. And therefore, we didn't pursue that one. But then what he did, that he started looking at how this tRNA is participating in ribosome biogenesis. And one of the things very quickly he found that if he had the wild type anticodon in the tRNA, which has mutation in 3GC base pair, it would cause biogenesis defect. So here we have the sector 3 and 4. You can see this one, that there is growth is weaker. Now, if he changes the anticodon from wild type to CUA such that it will not normally participate in initiation, that defect is not seen. So here is a situation where he has changed the anticodon, and sector 5, the growth is quite well. So he figured out that there is actually involvement of this tRNA in initiation, and that must be connected with ribosome biogenesis. Now the question was, is it really so? And then we went back to some of the earlier work that another graduate student, Smriti Arora, had done in the lab. And what she had done was, soon after Venki published this paper of crystal structure, how much time do I have? You have about 10 minutes. Oh, great. So what she found was that ribosomal protein S9, there are, this C-terminal tail of S9 is actually going and contacting the 3GC base pair. So we have the P site here, and this region right here is 3GC base pairs. This tail is contacting it. Also, there are a couple of methylations at 966 position and 967. These methylations also sort of make the contact with initiator tRNA a little bit tighter. So if we lose these methylations and lose the C-terminal tail of the S9 protein, by the way, this S9 tail 3 amino acids, SKR, are highly conserved. So when she deletes this, she finds that again, there is compromise in initiator tRNA binding, and in fact, there is cold sensitivity. We didn't connect it at that time, but Sunil looked at it, and he also then decided that he will try to combine his 3GC mutation with these mutations, and also with another thing, which is RBFA, which is a bona fide ribosome biogenesis factor with the 3GC, and this is what then he found. So he takes this S9 delta 3, and you can see that there is actually further defect in the growth. But he finds with RSMB and D, these are the methyl transferases which methylate 966 and 967 and 966, and you see that there is a compromise in the growth. But when he combines with the RBFA, there's no defect. And you can see that RBFA, you accumulate a lot of precursor. With RSMB and D, you start accumulating more. But with introduction of the 3GC base pair, there's really no change. So what this really meant was that the 3GC base pairs must be working downstream of RBFA. So when you have RBFA defect, 3GC is not coming in the picture. So 3GC role has to be downstream of RBFA. RBFA is working very early on. So 3GC must be working downstream. So then the other thing that he did was he then started looking at the combination with various RNases that are involved in processing of this 17S rRNA. And so many of these strains are available. So he just combined this 3GC plasmid he took into these strains, so that if there is actually a load, increased load on RNases, he would now see a processing defect, and he did see. So he sees in RNase B, R, and G, 3 of them. And that is shown in the growth also, that in the control, the strain is going like this, it goes flat. So this is one way of looking at that there are actually role connection between the 3GC base pair and RNase, and he can do a complementary experiment, and that is he can now overproduce RNases into this system where the 3GC plasmid is already there. And you can see here again that when he brings it at 22 degrees, here is the sort of control where he has the plasmid, 3GC plasmid, and the vector. One doesn't see much growth. But when he brings in the RNase PH, the growth starts to happen. So all of these RNases that we were talking about, RNase R, one can start seeing the growth. So again, there is a connection between 3GC and RNase R. Now the point is, the initiator tRNA is doing two things. We are saying that it's involved in biogenesis, but one can argue that we are affecting initiation of protein synthesis also, which means we might be depriving the cell of some ribosome biogenesis factor. Now, which one of these is correct? And what Sunil then did was he decided to do an experiment, old-fashioned experiment, and that is he took the cells, put rifampicin. Now, rifampicin is going to block any new transcription, and if he can see that the processing continues, that means in most likely situation that the new protein synthesis is not required. And this is precisely what he saw, that if you have here the control, so let's talk about these 15 minutes and 30-minute control, which are not treated. Now you treat with rifampicin and incubate them for 15 minutes and 30 minutes, the precursor levels keeps coming down. We can also incubate, while we are incubating the cells, the antibiotics that inhibit protein synthesis. This processing is not interfered with cosigamycin, streptomycin, chloramphenicol, fusidic acid, puromycin. None of them affects the processing. The only thing that affects is tetracycline. Now, tetracycline also is known to bind to ribosomal P site. So it seems to be competing with the initiator tRNA. So this then suggests that the initiator tRNA is doing a job which is more in the ribosome biogenesis than in the protein synthesis. Had that been the protein synthesis, we would not have seen this processing happening, especially in the presence of rifampicin and the antibiotics that inhibit protein synthesis. Then the question, of course, was that whether it is at the 30S level or at the 70S level. Now this, again, he did the experiment where he fractionated the ribosomes or the polysomes on sucrose density. We can isolate the peaks, 30S, 50S, 70S, and polysome, and now find out, again, which of these population accumulates more of the precursor 16S. And he found that in case of this particular strain where we are overproducing 3GC tRNA or the mutant tRNA, we accumulate more into the 70S. So here is the vector. You hardly accumulate any precursor in the 70S. With tRNA, wild-type tRNA overproduced, we don't accumulate much. But in this particular strain where the mutant initiator tRNA is being produced, we accumulate precursor 16S rRNA. So it is, and we've done a few other genetic experiments that I'll spare you with the details of those. But all of that then led us to propose a model which follows us like. So we have the precursor 16S rRNA in 30S. This is capable of binding initiator tRNA. It can be joined by 50S subunit. At this point, initiator tRNA does something which then allows the ribonucleases to clip off the final terminals of the 17S rRNA to give rise to the mature 16S rRNA and then go on to protein synthesis. So this would probably be happening only during the pioneering round of ribosome biogenesis. Now, the point that can be asked is, this is our completely artificial system. Would it have any relevance whatsoever to the in vivo situation? Now, the only way it can have any relevance would be if there are situations where the levels of initiator tRNA can be altered. So this, there are situations that are known which cyclic AMP cap, in fact, it binds to a downstream gene, ARG-G, but the promoter of RG is sort of overlapping, or the cyclic AMP binding site is overlapping with the promoter of MET-Y. And this binding actually downregulates. Again, you can see energy connection here. There is also a connection that the promoter of the gene, locus, which has 3 tRNA genes, is downregulated by PPGPP. There is a very interesting system that some of the viruses, in fact, come with endotoxins. WebC here, which is a ribonuclease, and this guy targets just the initiator tRNA, looks for 3GC base pair, targets that one. So there are situations in the cell which can downregulate the level of initiator tRNA. If that, then could the heterogeneity at the 3' end of the 16S rRNA be of any use to the cell? That is the question that arises. Now, the answer to this, we sort of started looking. There's a paper published by Harry Noller, and what he has shown was that you have in the 16S rRNA helix 28, helix 23. The anti-Shandel-Gano sequence is somewhere right there. When you make a pairing with the messenger RNA, these two helices actually sort of go away. And depending on the extent of the base pairing and the placing of the base pairing, one would imagine that this displacement would be different. Now, why am I talking about this displacement? There are a couple of residues here, 1338 and 1339, which are right next to this helix 28. And these two residues, 1338 and 1339, interact with the 3GC base pair. So there's going to be a direct connection there. So then a student in the lab began to change the extent of the Shandel-Gano and anti-Shandel-Gano sequence interaction. And what I can actually show you from this slide right here, the moment he goes into base pairing in the immature region, this whatever shown here in red is actually not present in the mature 16S. You can see that there is increase in protein synthesis with the mutant tRNA. And again, you can compare this versus this. This is completely mature. This is immature. There's increase. So it's quite possible, and of course we are looking for this now, that under stressful conditions, which let's say downregulate initiator tRNA in some way, there is heterogeneity that is created in the ribosome. And that heterogeneity is useful for the cell to, in fact, come out of those stressful conditions, and sort of, there is a change in the proteome, and that can be done. So, of course, a few other pictures that I found. Now, the acknowledgements, really, this work was begun by Suman Kapoor, Smriti Ahuja. Sunil Shetty really took it to a conclusion, and Riaz is continuing with this. And the laboratory is supported by funding from Government of India sources. And thank you very much for your patience.
Rajinder.
Site.
I'm sorry? So it's called as PI site. You know, this is the term coined by Jody Pugliese. So it's in PI site. It's not really oriented in a place where the acceptance time will find a position in the P site of the 50th.
So how does the tRNA then move to the real P site?
So that's where I believe the way it is happening is that the 3GC base pairs are interacting with G1338 and A1339. And that interaction is really what is involved in the conformational change, which allows this tRNA to go into the PP state. Well, both are connected anyway. Yeah.
I have a question. So
Yeah.
this was done in bacteria, correct?
Yes, yes.
So does this apply to mammalian cells?
Good question. The only thing I can tell you about that is that people have measured the levels of elongated tRNA and initiator tRNA under cancerous state, and I believe they find that the relative levels of initiator tRNA go down there. So I think this may
Actually, we
find some
found that
connection.
tRNA is double in cancer cells in general.
Initiator is more.
Yeah.
Okay, so there are some reports which I think but that again will be relevant from
Right.
change in the
Right. Anyway, any more questions? Yeah.
Sir, very nice work and very good presentation. Sir, you have done a lot of work on ribosome biogenesis.
some work, yeah.
Yeah. So my question is a kind of layman question. There are two kinds of ribosome, as we know, that eukaryotic and prokaryotic, and they are quite different. Even the ratio of RNA and protein are different. The prokaryotes have 60% RNA and 40% protein. Eukaryotic have 40% RNA and 60% protein.
I think you're talking about mitochondrial.
Yeah,
ribosomes.
this is what I'm
Yeah.
saying. So means within the same cell, we have these two kind of ribosomes, the prokaryotic and eukaryotic in cytoplasm and mitochondria. What is the need of having these such a complicated ribosomal systems in a cell?
That I will find difficult. I think Tom can only answer that. Tom. I think you require some of the proteins to be synthesized within mitochondria, and perhaps I think it's difficult to take them from cytosol to mitochondria. I can only speculate. So I think you need to make those proteins away from cytosol into mitochondria.
Thank you.
Okay, any other questions? All right, with that,
Thank you.
thank you so much. Oh, I'm sorry, one more.
It's not a question to Professor Varshney, but it's only a addition to the question asked previously. It's very clear the evolution of the organelles, mitochondria and chloroplast, and that tells you the answer.
Yeah.
Okay, with that, thank you very much, Mish. All right, our next speaker is Matthew Sachs. He's in the Department of Biology, Texas A&M University in the US. And he's going to continue this theme. So he's going to talk about gene regulation through the control of ribosome movement.
Right. Well, thank you, everybody, for attending, and thank you to the organizers for putting together such a beautiful conference. I'm going to tell you today about translation initiation in eukaryotes, and I will focus experimentally, of course, largely on work that has occurred in my laboratory and the laboratories of my collaborators over time. I think you can see here first the main subject of the research, which is the fungus Neurospora crassa, and you can see how rapidly it grows. This is time in hours. It grows millimeters per hour. And I show this slide because, in fact, can the lights be reduced is the question. Yeah, it is pretty bright in here. I agree, looking at. Okay, not a question. Is that better to see? Yes. So this is classic growth of a filamentous fungus. This is an Ascomycete, millimeters per hour. And you can see it's now sporulating. And in fact, when I was a first-year graduate student, Marcia talked about her decision to choose a lab. This is an image, almost another kind of a time lapse, right? This is probably the early 1980s, and just a close-up, of course, there's Gobind and Tom, and I joined Tom's lab in the basically 1980 because Stu Brody had published this paper, oh, don't do that, I don't think that's going to just slide on there, but okay. That the circadian rhythm in Neurospora, the 24-hour cycle, the sporulation cycle could be controlled by the mitochondrial F1-FO ATP synthase. And I was a new graduate student, and the question of big questions, solving the circadian rhythm seemed like a reasonable question. And Tom and Joyce Heckman had published a paper on Neurospora mitochondria, so it seemed obvious to me that I should go to Tom's lab and understand nuclear mitochondrial interactions and solve circadian rhythms. And in fact, my first, and many of you remember these reports who were in Tom and Gobin's lab. This is my first report typed on an IBM Selectric. And the words that are still here many years later, Neurospora crassa, in vitro translation, and N crassa mRNA and protein synthesis. So I probably have not strayed an initiator tRNA. They come together and they form this 43S pre-initiation complex, which loads at the cap. And here we're in the GTP-bound state. So we've loaded at the cap, and it's now a 48S because the mRNA is there. Just resuming here, we begin scanning. And I wanted to go into a little bit of detail here because of things that we say that turn out on close examination to be a little different than what we thought. Many of us even teach in biochemistry, GTP to GDP hydrolysis is the trigger for the reaction. What John Lorsch showed very beautifully, and Katsura Asano and I did a commentary on it, was that GTP and GDP plus PI exist in a metastable state in the scanning ribosome. And when an AUG is found, in this case an AUG, when an initiator is found, that triggers the release of EIF1, and then the phosphate leaves. And that's the trigger. So it's not that GTP hydrolysis is the trigger, the release of PI is the trigger. Some of us may say a very small point, but for some of us, it's a very big point. Yes. So this is a more modern view. This is from Alan Hinnebusch's paper, and I've just put our depictions underneath where we have, and there, where we have a 43S pre-initiation complex, loads on the RNA. And here, notice it's scanned past, in this open conformation, scanned past a UUG. And then when it finds the AUG, once again, EIF1 leaves, phosphate leaves, and now we move to a closed complex. And just taking that closed complex here, we then have subunit joining and initiation begins. So the first question is, how do you choose an initiation codon? And what I'll say here is Marilyn Kozak in the 1970s, 1980s, some of the original work showed that you have preferred initiation contexts, and these are often called Kozak consensus contexts. And overall, an A at -3 and a G at +4. And those of us who aren't computer biologists don't understand this, but biologists say +1 and -1 with no 0. right? So there's no 0 here. Just go from the +1 AUG to the -1. So it's terrible in some senses, but if you know it, you're fine. So if we look in Neurospora, it's very similar. So what happens, right? Many people who work in bacteria know that AUG is not the only initiation codon. But in eukaryotes, it's only now really widely appreciated that that's the case. So work a couple years ago we did where we basically put luciferase reporters with all of what are called the near-cognate initiation codons in to drive luciferase synthesis. So each of these differs from the ATG in an RNA code, the AUG, by a single nucleotide. And there's our control differing by 2. This lysine codon should not initiate. And with Ivaylo Ivanov, a longtime collaborator, and John Atkins, we also did very similar work in mammalian cells. And of course, what you see immediately is in a good initiation context, CUG is a pretty good initiator, right? Now, how often do we talk about CUG-initiated proteins? Not so often, and I'll return to that. But one thing I just want to draw your attention to here, if you change that second position of a near cognate to a purine, you're basically, as we would say humorously, dead meat, right? So if you're going to do mutagenesis of a start codon, you could change it to a stop codon, change it, put a second position change here. Otherwise, there's a good possibility for some synthesis. Okay, that's part 1, just to get a sense of initiation. Part 2 is, and certainly in 1980, and the idea was that eukaryotic mRNAs have a single AUG codon and a single reading frame. Now we know upwards of an order of 50% of mRNAs have short upstream open reading frames. And their purposes are multi or not at all. In the fungi, we're at about the 22% range, and these are AUG initiated. So what do these do? Obviously, they could have coding capacity, but they certainly have regulatory capacity. First, because of the nature of scanning, you could simply reduce downstream initiation by putting in some upstream initiation. So I'll simply call this preemptive initiation. If you initiate here and you don't have an efficient reinitiation mechanism, and we'll come to that, then basically you reduce downstream gene expression. That's kind of like the de facto one way it could work. I spent a good 20 years studying ribosome stalling at an upstream open reading frame termination codon. This is a regulatory stall. And I'm going to show you some of these, some of the data for this, and some of how now we can use evolutionary conservation to maybe be able to hone in on what URFs, I'll just call them morphs, what URFs have that could have this capacity. Genetics in Saccharomyces in the 1970s, and my group's work in the 1990s, asking for the loss of arginine inhibition of the first enzyme in arginine biosynthesis's translation, well, actually just its expression, revealed an upstream open reading frame, and that a mutation at this aspartic acid in either of them removed arginine-specific negative control. First gene in arginine biosynthesis, not enzyme feedback inhibition, but literally shutting down gene expression. It turns out this is through the ribosome stall, and that ribosome stall also triggers nonsense-mediated mRNA decay. Those of us of a certain age remember attenuation. This is, in a sense, like attenuation, but it's at the level of RNA stability instead of allowing transcription and elongation to continue. So historically, 1944, which is 3 years after Beadle and Tatum's 1941 paper, the ARG2 gene was discovered. And I like to show this slide because it says in the abstract that for studies of intermediary metabolism, the new science of genetics furnishes a very powerful method. Now, 1940s, I know for some of us in the audience, might as well have been, a long time ago, but it isn't. It's, and in fact, this is shown now, and now, of course, we think of systems biology, proteomics, and genomics as very powerful methods. So here we have the model for how the stalling, low arginine versus high arginine, triggers does two things. It stops ribosomes from getting to here. And I want to point out that this initiation codon is in a very poor context. So most ribosomes go by it. But if they find it, then, and there's arginine in the cytoplasm, then they stall. And now, if you simply, instead of doing alanine scanning mutagenesis, you look over 1 billion years of evolution, the chytrids and the Pizizomycotina, which include Neurospora, you see this very strongly conserved region of these peptides. So for 1 billion years, it's been like this. And if you do the alanine and proline scanning, this is the region that's important. And what you can see, hopefully we will see, is how this peptide acts within the ribosome to make a stall. What I can say is biochemically, we know that arginine itself, and even an RGD tripeptide, can get into a ribosome, a stalled ribosome structure, and trigger a conformational change in the peptide in the ribosome that interferes with the activity of the peptidyltransferase center. So this is a fairly well worked out mechanism for stalling. Now, going not too far forward, inositol, another one of these metabolites, right, of critical importance, 1944, the first inositol biosynthetic gene, which of course is the most important step in inositol biosynthesis, inositol 3-phosphate synthase in Neurospora. Ivalo and I were looking at these sequences, and we saw that there was an upstream open reading frame initiated not with an AUG, but an ACG. And if, again, we look over 1 billion years of evolution, it's highly conserved in the fungi. Not Saccharomyces cerevisiae, but in the other fungi, some of the other Saccharomycotina. And I'm going to come back to these histidine codon, and I'm going to come back to this leucine 6. So what could this be doing? I already showed you with arginine biosynthesis that we could more or less show, using in vitro translation, that it stalled ribosomes. And first of all, just once again, it's in all of the fungi, what we call the classic fungi, not in animals. In this age of translational research, I'd like to think that that still wouldn't matter, but we will know when we know. But if we look at the start codons, once again, they're a variety of near cognate codons. And in the organisms where it is an AUG, it's actually a very poor context Kozak sequence. So there's a U at -3 instead of this important A. So presumably the ribosomes don't often initiate, but when they initiate, our model is that they would stall the ribosomes. And many of you are aware of this technique called ribosome profiling, where you can map the positions of ribosomes on mRNAs. And of course, we've done that. And if you look at this inositol RNA, there's the coding region. That's the 3' region. There's not many ribosomes there. But in the 5', we call it the UTR, the untranslated region. But of course, it's translated. But we're just going to stick with the UTR nomenclature anyway. You can see that it is translated. And we actually see some evidence for what possibly is internal stalling. But we'll return to that. But it's clearly translated in living cells. Just some controls here. There's the arginine attenuator peptide UORF. And I'm not going to talk about this, but EIF5, one of those critical initiation factors, is auto-regulated, and it has 2 upstream open reading frames that we've demonstrated are critical for its regulation. Well, okay. So there now we've fused inositol directly to luciferase and looked in vitro. And what we have shown now is, especially if you look at a good initiator, that you can see an inositol-dependent loss of luciferase synthesis. This is adding just inositol direct. Okay? If we do what's called primer extension inhibition, where instead of making a full-length cDNA, where the ribosomes are located, it blocks expression, we can see, in the case of the arginine attenuator peptide, we can see this arginine-dependent stall that's absent in the D12N mutant. And notice if you shorten the upstream open reading frame, you lose regulation. The sequence length is critical. And if we look at the inositol peptide, we can see the stall here. And it looks like a stall during elongation. These are the mutants that I pointed out, the his double and the alanine mutant. They no longer show this stalling. So we think what's going on here is that inositol is stalling the ribosome. And this is now another example of a small molecule metabolite controlling translation. Finally, reinitiation, and I'll have to speed up a little bit, where you have the ribosome actually not falling off, but reinitiating. The classic example of this is cross-pathway control GCN4, which is a BZIP protein. And in Saccharomyces, many people are familiar with GCN4 and its control. And ultimately, what we see with GCN4 is the ribosomes choose to initiate at URF. They all initiate at URF1. They initiate at 4 or downstream. And we thought that Neurospora was going to be just the same as GCN4. So we did some in vitro experiments, and we saw a larger band, 35S-methionine, larger band. I won't go through all this. This just shows that the URFs work like they ought to. We were puzzled by this larger band. So when we looked, we saw that this reading frame for CPC1 was open. There wasn't a single stop codon in this entire reading frame in over 100 different piezoomycotina. And furthermore, we saw when we did the ribosome profiling that there were lots of signals there in the same frame. And furthermore, when we did the evolutionary analysis, we saw this is the main AUG here, and that's the conservation. There's just as much conservation upstream. And there's also other examples unproven yet in the fungi for CUG initiation. So to wrap this up, then, what do we want to say? AUG is not the only initiator, right? We can diversify protein functions, we can increase regulatory capacity, and probably for everybody in the audience, new challenges for functional annotation, right? And in the interest of time, I won't go into too much detail about elongation, but you can control elongation, you can control frame shifting, you can basically control codon preference matters, and you can control elongation factor phosphorylation and control rates of movement. Termination, I just showed you some examples of termination. Programmed suppression is crucial for retrovirus translation. There's examples where you can make targeting for proxysomes by programmed read-through of a stop codon. And of course, there's RNA quality control mechanisms that sense premature termination. So let me conclude by acknowledging people in my lab and at Texas A&M who have been helpful, especially I work a lot now with Deb L. Peterson, Ivanov and Tom Deaver. We're working together on the inositol project. And with that, I need to stop and take questions. Thank you.
Okay, thank you so much. I'm actually going to start with a question, which is you mentioned inositol regulation, metabolite regulation. I presume that's to coordinate the ability of the cell to kind of respond to the environment, what nutrients are there, and then how many proteins you make in response to that, or how you basically use those nutrients for different aspects of metabolism. My question is, why inositol as opposed to something else?
Yes, I asked this question. The question is, why inositol? Why do this? And I asked this question about arginine because the cell, that question I have a better answer to, I think.
Okay.
Because fungal cells will put 1 molar arginine into their vacuole, even in minimal medium. And in fact, the trigger is about 100 micromolar that we see. And that is about the cytoplasmic level of arginine. So presumably it's a homeostatic mechanism that if you have 100 micromolar arginine in the cell, you're happy and you just degrade the message that you made. But if you have less, then you're stabilized. So presumably with inositol, it would be a similar kind of mechanism, not an external sensor, but an internal sensor. That's my idea. Whether it's true or not, I don't have an answer. But you can certainly force, like you were saying about putting in unnatural amino acids, you can put arginine in from the outside and see an effect, but I don't think that's what it's there for.
I had a question as well. So these upstream ORFs, I think you didn't mention much the proteins that they may encode. And
yes, the dark matter of the universe, the proteins they encode. Yes, and Takagaki and I were talking about this a little bit. You know, historically, we only annotate proteins that are larger than 100 amino acids. Right. Yeah. So far as we know, these proteins, we don't have an identified genetic function, but there are multiple examples now of UORF encoded proteins that have physiological functions besides acting as cis-acting regulators of the downstream translation unit.
So that's something for the future then, new methods to analyze that.
Yes.
Because computer algorithms won't do it. I mean, there's not enough signal, I guess, to annotate those reliably.
That's a great question too. So what's happening now is with respect to annotation, and many groups are using the ribosome profiling data to at least get some kind of truth statement with respect to whether or not there's evidence for their translation, as opposed to just their existence as an upstream AUG. But yes, it's annotation issues.
Yep.
Here. I'm not sure where here is.
Here.
Okay, yes.
All right. So actually, I just wanted to share something and then wanted to ask a question. Like, you know, if you take a simple 100 amino acid protein and you know, if you make all the permutations and combinations with all the available amino acids, you're going to have a 20 power 100 combinations of proteins. That's a very small, you know, protein. We have got 1,000 amino acid proteins as well. So, we just have, you know, approximately, you know, 10 power 80 atoms in the universe. I mean, so I mean, how, you know, only if you look at all the combinations available, in about 6 lakh proteins, one protein is in existence. Not all the combinations are made on Earth, actually. So can you relate this thing with, you know, the preferences of, you know, translation over some codons, and why only specific proteins have come into existence, not all? Had it been a stochastic mechanism, then why all the proteins did not come?
Oh yeah, it is a great question because we tend not to ask these kinds of questions, right? And it's the same way. I mean, and I'm just going to, of course, change it into my question, which is probably not fair. But you know, when you think, the question of big questions, right? And Marcia kind of also got a billion years, why have fungi wanted to sense inositol in this way, and how could you even get such a sensing mechanism? One of the arguments for URF evolution is not a positive one. It's that if you get an AUG mutation in a 5' UTR, then you've got problems because you're no longer making your protein, because you're initiating upstream, And if you make a long protein, you don't reinitiate. So the idea is you get stop codons as the simplest way to suppress that upstream AUG. But then somehow from there, now you have to select a structure that can bind arginine. I mean, that's not so obvious. So even with the small UORFs, which is a shorter version of your problem, right, I think now the way the field is looking is to look at the ones that have evolutionarily conserved sequence elements to at least try to understand what those domains might do.
How about saying that they might have come, but not selected?
Actually,
they might have come, but
they would have come. I mean, I mean,
they might have come, but not selected.
Because
right. Yes. Yes. So it's the retention.
Yeah.
It's a retention, right? So in fact, that was a question we had for the part that I did go just 22 minutes, I might add, so I'm okay. But I did speed up a little bit, and I went over it quickly. That was an important part for the CPC1 when we were looking at that upstream thing. I didn't show you that it's published now, but each of those near cognate initiators isn't only one. There's like 6 of them, and you can knock out one, and then you see other forms. And so that was a question, right? I mean, if you do the synonymous versus non-synonymous DNDS calculations, at least for that region, we see evidence that it is being maintained as peptide sequence, and that's the best we can do now.
Selection.
Yeah, there is some selection for those.
By
yes.
niche.
Yeah. Yeah.
However, if you look at the peptide transfer center, in the 50 angstrom radius, there is no peptide at the peptide transfer center. So I just want to know that, why are we not talking much about, the 23S ribosomal RNA regulation?
Right. Why am I not talking about it? Because we've actually done work with groups working on TNAC, which is the E. coli tryptophanase leader peptide that stalls ribosomes in response to tryptophan, using Kathy Squire's Delta 7 rRNA strain, where we could select and make mutants in that region of the ribosomal RNA. So we could look very specifically at the rRNA universe. The problem is with the nucleolus and multiple copies of the rRNA, some yeast workers are now have figured out how to do that, but we haven't in Neurospora at this point. But yes, that is a great space to look at. The problem is multiple copies of the rRNA kind of mitigate against that.
I mean, just a small thing. If you look at the proteins of ribosomes, and if you break it down into smaller peptides, they are just mere repetitions of small peptides, and those peptides are also found in the normal proteins of the cell. If you see, I mean, how the protein got a three-dimensional structure would be a repetition of these smaller peptides, and the small peptides won't have any three-dimensional structure as such. So, yeah.
Okay, so I'm not sure I fully understand your question, but one aspect of these peptides is that they are cis-active only during synthesis. So there's a very high local concentration. It's stoichiometric there at that moment. And that was a little bit about Nico's question earlier. These are cis-acting regulatory peptides. These are not diffusible transacting peptides, because that's not an explanation, but that's a rationale for.
The interesting thing is still ribosome is still the fossil of the RNA world. We don't have any protein as such which is able to synthesize protein. They still have to depend on RNA.
RNA rules. We all know.
Yes. That's a fair statement.
No, I'm going to pass. I'm going to pass in the interest of time. Yeah.
Okay. Well, good.
Okay, well, let's thank Matthew. All right, so it's my very great pleasure to end this session by introducing Tom Rajbandari. Someone asked me why we call him Tom instead of Uttam. I don't know, but that's what he told us to call him. From the last two talks, the elegance last two talks, you can see his great influence on science. And as we've all commented, Tom is so modest, he never really talks about his own accomplishments, but they've been extraordinary, and he's been really a central part of the Khurana Rajmundari legacy. Because it's Tom, I'm going to let you talk as long as you want. So here we go.
Let me see. Where is this? Okay. Okay. Great. Yeah. Thank you, Marcia. I needed that. This morning, Dieter Sol, who I have known for close to 60 years, reminded me that I'm the oldest person here at this meeting. Yeah. He's only 6 months or 4 months younger than me. So, I went to, when I joined Govind's lab, it was October 1, 1962. I think it was a Friday, and arrangement was that I would go visit Govind on Monday so we could talk about what I was going to do, because he wouldn't let me know beforehand what I was going to do. And on Saturday or Sunday, I went out desperately looking for a place to get something to eat. And it was some kind of a small drugstore that also sold food. And the person sitting at the place was Dieter Sol. So he had also just arrived maybe a month before, whatever. Yeah. And the interesting thing is that was not the first time I met Dieter. I had met him at the place where I did my PhD. And so Dieter was spending a month, not month, a year, because all German PhDs have to spend a year in another institution, and he was spending the year at the same lab where I was doing my PhD. I was going to do my PhD. Yeah. So that's how I know Dieter. Right. So I met Govind, and he told me what I was going to work on. He was going to give me a project that he himself was working on, because he was going to move on to better things like the genetic code. This was 1962, right? So that what I got from Govind was that I would work on some way of putting tags on the ends of nucleic acids. Nothing was known about nucleic acid sequence, nothing. Some way of putting tags on the ends of nucleic acid sequence such that we could begin to get some information. In much the same way as Fred Sanger put tags on ends of proteins or peptides, and with DNP and started getting sequence information of peptides. So basically same principle. So he handed me his notebook. And I looked at the notebook. When I went home, I looked at the notebook, and it frightened me. It frightened me because Govind is a meticulous note keeper. He wrote, he did an experiment, right? And he put the time, right? And what the results were. And so one day, notes would read like 6:30 p.m. Finish this, put samples on the counter to count, went home to eat dinner. And then it would start with 8:30 p.m. same night. Yeah. And then he described whatever he did until about 11:30. And then the morning would begin. So something like that. So that really frightened me, because it basically told me what the expectations would be, without actually telling me that I was expected to do that. So that's really, but it was a wonderful thing, of course, opportunity. And so this is what I was doing, basically. People talked about coenzyme A yesterday, how John Moffatt synthesized coenzyme A. John Moffatt synthesized coenzyme A by using something called phosphoromorpholidate, right? So what I was supposed to make this compound here, methyl phosphoromorpholidate, and react it with that phosphate here, and that way put a tag, that with C14 label tag. And then if that reaction worked, and then I would isolate that, and then I would cut it at this site, and isolate the radioactively labeled nucleotide and identify the nucleotide. So this was the idea. So I did that. After a while, I was succeeded. I did that on a total tRNA of the cell. So it was a mixture of tRNAs. The idea was nothing wasn't known. So really was, I was interested in doing that. So and it was known that there was a lot of G there at the end, which is what I found. But when I did a two-dimensional chromatography, I found there were 5 spots. 4 of them were in A, C, U, and G, as expected, and a 5th one, which remained unidentified until later, that turned out to be pseudouridine. So this was the beginning of my work at Wisconsin. And then we talked about what to do next. Well, even before we published the paper, then we talked about what to do next. And it was after about a year after I arrived there, I think. So we said, I think people are getting interested in tRNAs because tRNAs are obviously very important. So the idea was now that we would now start purifying tRNAs and actually see if we could sequence a tRNA. So that was my job. And Govind was incredibly generous to me. He gave me 3 postdocs and 2 technicians, and I had a lab downstairs, right? And so 3 postdocs, 2 technicians, and just left me alone, literally, except to go upstairs and go to the seminars and do things like that. So that was, and then as I was trying to sequence tRNAs, paper came out from Bob Hawley's lab, who had actually sequenced the tRNA. 1965, he got a Nobel Prize for that. I shared it with Govind. Right. So anyway, we continued with it because so many tRNAs, and that was not the end of the world. So we were working on a different tRNA, and we sequenced the tRNA, and that turned out to be the phenylalanine tRNA of yeast. So this was a very, very useful tRNA, really, because people really started using this tRNA for all kinds of reactions, all kinds of purposes. It was the first tRNA that was crystallized by Alex Rich in Cambridge at MIT, and by Aaron Klug at the MRC labs in Cambridge, in England. And so that was a very useful thing. But this tRNA was interesting. It shows it's actually 76 nucleotides long. It contains 14 different modified bases. And this is really a lot of modified bases. And with this tRNA, then we could begin to do experiments now, biological experiments. And the first, and again, the Govind's lab had a lot of collaboration going on between people. And the person I would collaborate, for first collaborative experiment I did was tizosol. So what was happening was, so this tRNA is interesting, right? The sequence, it has the anticodon sequence GAA, right? And Francis Crick in 1966, we sequenced this tRNA in 1966. We reported partial sequence at the Cold Spring Harbor, published this paper in 1967. And then Francis Crick had meanwhile published some idea called wobble hypothesis, which said that you do not need a perfect pairing between the messenger RNA codon and the anticodon of this tRNA. There is some wobble. Wobble means some, you can have more than just perfect hydrogen bonds. That if you have a G here, that G in the anticodon can pair with either U or C, and therefore read 2 different codons for the same amino acid. So that was just a hypothesis. And we here with this purified tRNA now, we could do, and protein synthesis experiments going on in Govind's lab, and Dieter's expertise in it, we could now do the experiment, right? And so we did the experiment, which actually proved that Crick was right, at least in that particular prediction. Okay, so this is the experiment where we now took that tRNA, we attached the radioactive amino acid phenylalanine to that purified tRNA, right? And then we added it to a protein synthesis system, and we asked, does it read both the poly UUU and UUC codons on in the translation system? And you can see that the radioactivity present in the tRNA goes into protein polyphenylalanine with both poly-U and poly-UUC. So this single tRNA can read both the UUU and the UUC codons, the first proof of wobble hypothesis. So that was one collaboration. And then we were collaborating with others in the process of doing a completely, innocent experiment. Really, it's just a control experiment. We came across an enzyme which is now called peptidyl tRNA hydrolase. And that paper we published with Hans Kossel, who was another guy in the lab, doing something completely different. So that was this experiment here, where you attach a radioactive amino acid to a tRNA and modify that radioactive amino acid. The amino group is modified with an acetyl group, so that you have acetyl amino acid attached to the tRNA. Or you can have a short peptide attached to the tRNA. And then you treat it with a cell extract. All right? And what you see is that all of a sudden the radioactivity just goes. So there's an enzyme in the cell extract that is removing that acetyl amino acid, right? And then we know that the tRNA is left intact because it can now be charged with amino acyl tRNA synthetase. So the amino acid can be put back in. So this was really the discovery of peptidyl tRNA hydrolase that Umesh continues to use still. So this was another really useful collaboration. And then we became interested in initiation of protein synthesis. And at that time, there was a paper that came out from Cambridge in England, which said that the initiating tRNA in E. coli is the formyl methionine tRNA. It's the tRNA that's charged with methionine and then formulated. Umesh mentioned that before, right? And so the question was, what happens in eukaryotes? So that was interesting. So we thought what I would do is we would just see if we can take a yeast tRNA and see whether there's any yeast tRNA that can be amino acylated with methionine and be formulated, thinking that maybe that would be also the tRNA used for initiation in a eukaryotic system. Right? So that was the idea, and we did that. And indeed, if we take a yeast tRNA, by that time we also fractionate this yeast tRNAs into two types of methionine tRNAs. And if we took those tRNAs and added amino acid, and then added radioactive formyl tetrahydrofolate, formyl group radioactively labeled, what you see is that with one yeast tRNA species, the formyl group goes in. With the other yeast tRNA species, the methionine tRNA species, it does not. One methionine tRNA, there is no formyl group incorporated. The other methionine tRNA, there is formyl group incorporated. So that said that indeed in yeast also there is a tRNA which can be actually amino acylated with methionine by using an E. coli enzyme, and then the E. coli enzyme will also then add a formyl group to it. So to us, this meant that maybe this is the tRNA that is used for initiation of protein synthesis in eukaryotes, about which nothing was known then. Right? So we became interested in it, and I think at that time we started doing protein sequencing the tRNA. In those days, each sequence of each tRNA took about 2 years, literally 2 years, a lot of effort. That's why 3 postdocs and 2 technicians. And then we sequenced it, and then I moved to MIT in Cambridge. And then the first thing we did, first paper I published from there, was a paper in collaboration with Harvey Lodish, done by one of his students, who is now a very well-known genetics researcher, also a faculty member in our department. And we showed that this tRNA, indeed, which is the so-called this formulatable yeast tRNA, the putative initiator tRNA, was actually initiating protein synthesis in yeast, which said that in eukaryotes also, protein synthesis is initiated with a special methionine tRNA, but not, it turns out, with formyl methionine. You do not have to formulate it, as we all know now. So this was really the beginning. And then the tRNAs became something that we really, just very interested in important things to do generally with protein RNA recognition processes and things like that. And I think for the rest of my life, I have stayed literally very, very close to the tRNA field. We got sidetracked on working with mitochondria for about 15 years. Matthew mentioned that, right? So, all right. So now, what I'm going to tell you today is really something simple, and it's nothing complicated, and just something that we've done in the last few years, and that has to do with basically the biology of tRNA in archaea. Now, archaea is, as you know, there are three kingdoms: bacteria, eukaryotes, and archaea. And this archaea is what we will focus on. And I'm going to really talk about two different aspects of archaea. One is how the genetic code is read in archaea, and the second one is why there are no nonsense suppressors that have been found in archaea. You might be, it's just not something that people know. Is that in bacteria, nonsense suppressors play such a critical role in genetics of bacteria and viruses. In eukaryotes also, nonsense suppressors have played a very critical role in genetics, in yeasts and worms. And yet, in this third kingdom, archaea, nobody has come across any nonsense suppressors ever. The question is, why not? So those are really two things I would like to talk about. All right? So, new insights into reading of isoleucine codons in bacteria and archaea. Why is this interesting? This is the genetic code. Now, this is a little different from Dieter. Dieter has given you the very latest version of the genetic code, a lot of his very beautiful work. Right here, I'm showing you the old genetic code. Right, this is the genetic code that is, to me, is very beautiful. Yeah, looks so well organized. It consists of 16 4-codon boxes, 64 codons, right? And it is you actually it belongs to two sets. One set, you have all four, one set, you have all 4 codons specifying the same amino acid in yellow, right? And the other set specifying, so there are 8 of those 4 codon boxes. And then the other set specifying where you have each 4 codon box is split into two, the 2 with pyrimidines specifying one amino acid, and 2 with purines specifying a different amino acid. And there's 6 such boxes. So the 14 codon boxes then are like that. And then the 15th one is here. 2 of them, again, specifying one amino acid. But the other 2, one is a stop signal, and the other one is a tryptophan signal. So that's also, okay, so now, this is, right, so according to Wobble hypothesis, a single tRNA with G in the anticodon can read any codons ending in C or U. And a single tRNA with U in anticodon can read codons ending with A or G. Right? So if we go by that rule, then we can explain basically how all of these codons can be read by sets of tRNAs, but without any ambiguity in the process. But here, with this system here, this one exception here is that you have a 4 codon box split into 3 and 1. Now, you can imagine that these 2 codons would be read by a tRNA, and this codon would be read by a U. But if that does, what prevents it from also reading that G? And if it does read that G, then it's a mistake, and that cannot be allowed. So there has to be a system here, right, that somehow there's a system that allows you to read the 3 codons for an isoleucine without also one of the tRNAs also misreading the AUG, which is a methionine codon. Now, there is some information on that. In bacteria, it is known that you have the U and C is read by a tRNA that has G in the anticodon, and the A in the tRNA is read by a compound that is a modified C. A C to which a lysine is attached, right? And that's called lysidine because lysine is attached to it. Exactly how that happens, how that reads a C, how that reads an A specifically without also mispairing with G and reading AUG is not known. Okay, so we did some work. We decided now what is the story with archaea, and we have found that in archaea also, it's the C, modified C, but the modification is different. You have something called agmatidine. We call that thing agmatidine because it's in bacteria you have lysine attached to it. In archaea you have agmatine. Agmatine is a decarboxyarginine attached to it. So, okay, so okay, so this is it. Now, all right. So this is the story here now. In both cases, you have one tRNA that reads AUU, AUC for isoleucine, and here a second tRNA with a modified C that reads A without also reading AUG. Okay, and so the question really is, how does C base pair with A? It's counterintuitive, right? How does C base pair with A and not with G? Pair with A without base pairing with G, right? And so the question is that correct? So I will just show you here, this is agmatidine, and this is lysine. Lysine, and this is agmatidine, right? Okay, so the answer came from Venki's lab after some effort. This is the system that is used. Is that you have the ribosome, you have a very simple message, AUA, AUA, right? And then you have your tRNA that we have sent him, and analysis. The results were very surprising. The surprising result was that, first of all, there's only a single hydrogen bond linking C to the A. So this is it, a single hydrogen bond linking C to the A. And really perplexing, how can that happen, right? And secondly, answer was that it actually there's another hydrogen bond between the long agmatine side chain. There's a hydrogen bond donor and hydrogen bond to the phosphate of the messenger RNA. So you have that is anchoring that there, and just a single hydrogen bond that is enough for the C to recognize an A. So that looked good. You still did not explain why C does not base pair with G. And then the answers came. I don't think this tells you much, but let's go here. That if you now try to build a model with G there, what happens is there's a serious steric clash between this long side chain and this the side chain of G. A group called G. So that is really what it makes, preserving its specificity for A without allowing it to base pair with G. So that was, to us, was a reasonably satisfying answer, and that's where things stand. Okay. All right. So I think that's really good enough. Now, so then we asked, okay, we got answer, right? We have, but why? Why do we have a system that uses a modified C to base pair with A and not a U, and just put some restrictions in it so that U can base pair with A easily? So the thing was we wanted to now see if we mutate the C of the tRNA to a U, what happens? Can we isolate the tRNA and see what its base pairing potential is, right? And we did that, and this is just show that it's purified and all that stuff, and we do all the analysis, and the answer was that indeed the tRNA, the wild type tRNA with agmatidine base pairs exclusively with A, but not with any of the other three isoleucine codons, right? But all with AUG. But the mutant tRNA with a U here still base pairs very well with AUA, but it has the potential for mispairing with AUG. That is in fact suggests that indeed, if you put U there, you have the potential for misincorporations of things. So that is where it stood, and we also characterized the U that was there, and it turns out to be also modified. And modified U is here. This is the analysis. These are the 4 markers, and the compound that we found is that. And that compound happens to be something called cyanomethyl U. They're not important, but but it's kind of explained to us why nature has not used a U at that site and used this bizarre way of modifying a cytidine, right, to base pair specifically with A without allowing it to base pair with G. All right, so that's number 1. So I'll go on to Marcia. You have to tell me when I need to quit. All right? Okay. Okay. All right. Great. Okay. I'm glad. Yeah. Okay. So nonsense suppressor in archaea. I mentioned that suppressors have played such a critical role in bacteria and eukaryotic genetics. And if we could have suppressors in archaea, maybe that would also facilitate archaeal genetics. And so that was the reason why we asked why are there no suppressors in archaea? And the question we asked is, well, can we just express a suppressor tRNA in archaea and see whether it will work in suppressor? Whether it is something that suppressors just don't work, or suppressors are inherently very toxic to archaea and things like all those are possibilities. So people in the lab took 2 different tRNAs, mutated the anticodon in such a way that you could get amber, ochre, or opal suppressors. They would read UAG, UAA, and UGA codons, right? From serine and tyrosine, just in case one is a strong suppressor and one is not, and all that stuff. So just to make sure that we get some positive results, basically. And so you put that into the cells along with a reporter system that allows you to see what's happening here. And indeed, this is just this expression system. I don't think we're interested in it. We can either have it a constitutively expressed tRNA or inducible system to express it, just in case the tRNA really happens to be toxic. And indeed, for this now beta-galactosidase is being used as a reporter. And you can see that if we put the beta-galactosidase with an amber mutation in there and also express an amber suppressor tRNA, you get suppression. So it says that there's nothing wrong with archaea. Archaea will actually allow suppressor tRNAs to function. And this is a plate assay here. And if we put an ochre suppressor will also read the amber codon to a small extent. And this is, again, similar to the in what we know in bacteria, is that in bacteria, the ochre suppressors, inducible system, and all that. And with that, we are all in business. And we also showed that actually you can have you can also correct or suppress oxytropic mutations in the cell. This is a PIR2 amber is being suppressed, right? Yeah, you can grow this here. This is with a wild type PIR2 amber with the 3 millimolar tryptophan index there is suppressed. And if you use wild type tRNA, it is not. Amber suppressor tRNA it is. So it says it's inducible, and you can get oxytropic mutants being suppressed and all that stuff. And now the interesting thing is that the level of expression of the tRNA depends very much on the site where we are expressing it from, on the chromosome. If we use a beta-gal locus, and if we just look at amber suppressor derived from tyrosine, serine, or tyrosine, you get similar levels of suppression, and inducible or constitutive, about the same levels of suppression. But if we now go into another locus, okay, so this is, okay. If we go into another locus, we can see that we get on the beta-gal locus, about the same as before we showed, but on another hydro B locus, we get substantially increased amounts of suppression. So that has become really extremely useful for us in the sense that now we can, some suppressors are so weak that we will not get any suppression otherwise. But if we can regulate it to such an extent, we will. And here I show the results here. In the inducible system now, we get, with serine, we get 38%. Tyrosine, we get 73%. Glutamine happened to be an extremely poor suppressor. We got 0 results on the beta-gal locus, but here we're seeing now suppression here. So you can, even if you have a weak suppressor, you'll get suppression. And similarly, now with hydro B locus, if we have amber suppressor, you get that. If we have ochre suppressor, you still get suppression. Opal suppressor, we still get suppression. So you can get a wide range of suppressants, amber, ochre, opal, with amino acids inserted being serine could be one, tyrosine could be one, glutamine could be another, and so on and so forth. So this is really where things stand. We have these systems. They're good, and we have kind of shipped it to 2 different labs, one in Europe and one in the States, so for keeping and just so that it can be safely stored. So that's really where things stand. And I don't have any much to say. Thank you.
So again, you can get old, not crazy, and do great science. question. Dieter.
Oh, okay, no, okay, 1 minute.
Okay.
I'll give 1 minute.
I really would like to make a comment, and I only make it here because there are many young people, and this goes back to initiation in E. coli. Haragosh is not here, but Haragosh and I worked and initiated here on E. coli. We submitted a paper, Gobin submitted a paper to then the top journal, Journal of Molecular Biology. We did not get an answer for over 9 months. While at the same time, a paper from Cambridge what came out of Nature.
Yeah, yeah.
Yeah, okay. Do we have any questions or comments? Yeah.
I have a...
Osim.
I want to follow up on my question from yesterday.
Jeez.
So, you didn't answer my question yesterday, so I'm asking it again. Umesh, please help me. People hold him down. Yes. We would like to start.
I already said we are going to do it.
He has to agree.
Well, he agreed.
Would you please agree to start a fellowship called the Uttam Raj Bhandari Fellowship?
You will never get a yes from him.
Wait, Shanti. Will you agree to let us start a fellowship in your name?
I'll give you the same answer I gave you yesterday, which is that I always listen to you.
I heard yes.
I always listen to you. Yeah.
That's a yes.
Yes, it says yes. Dear, I have a comment.
Yeah.
How true. We submitted a paper to the Cell. The people from Cambridge had already submitted a Cell-out paper over there too. He sent it back. He says too specialized for us, but they were already reviewing on the other end.
And you see, the small selective journal that we founded now has a better impact factor than the proceedings of the National Academy of Sciences.
Okay, yeah.